It's Biology with Mr. B. Biology with Mr. B. That's me. Good morning, peeps. So, today, I want to get straight on in with kind of the remainder of the spec point B, because hopefully yesterday you've looked at the lac operon, which was genetic control at a transcriptional level, but for prokaryotes. So what I want to look at today is, well, what does it look like for organisms that don't have operons in the same way prokaryotes do? So what does it look like in eukaryotes, transcriptionally? And then what does it mean post-transcriptional? And what does it mean translational? How can you control a gene after the protein's been made? That's what that's saying. Now here's the good news. Because we've done the lac operon, and we've got this idea of a promoter region, operator region, the eukaryotic control is actually quite straightforward, really. And we can actually link it really nicely to steroid hormones and how they work. So we've actually kind of already had a look at that. Post-transcriptional is new, but again, you should find it quite nice. We're just going to imagine a nice pair of scissors. A nice pair of scissors on the messenger RNA. Because post-transcriptional means the messenger RNA has been made, but it hasn't yet been transcribed at the ribosome. So maybe you've just made the mRNA and it's still in the nucleus, it's yet to leave the nuclear envelope. Translational is, we have a protein. So what can we do to either make that protein work or maybe inhibit it? And that would still count as gene control, even though you're doing it on the protein that's already... I know it's a bit confusing, but still does count an element of gene control. So anyway, genes can be switched off. That's the key message. I mean, we saw that yesterday with the lac operon. In the presence of glucose, and when there's no lactose, but in the presence of glucose, that repressor protein that was made literally switched off the beta galactosidase and lactose permease by binding to the operator region on the operon. And it meant the RNA polymerase just could not bind to that promoter region. So those structural genes, LAC-Y and LAC-Z, were switched off. And this is important. I mean, every single cell in your body, every single nucleus contains the exact same genome. We are a product of mitosis from that early zygote. Every single nucleus is identical. I don't need every single gene to be switched on. I don't need the gene for salivary amylase to be switched on in my foot. I don't want my feet to salivate. I really don't. So how do our cells control which genes are expressed in eukaryotes? What starts protein synthesis? Well, the answer is a little protein, or a short non-coding piece of RNA called a transcription factor. Transcription factors, these little proteins, or sometimes short non-coding pieces of RNA, they are able to act within the nucleus to control whether a gene is turned on or turned off. And what they do, these are little proteins or little non-coding pieces of RNA that are involved in attaching RNA polymerase to your DNA. So they've got pretty important parts, don't they? Now, there are loads of different transcription factors, and for each specific process, there are lots of different transcription factors all involved. They all have a different shape, a different sort of tertiary structure, in particular if they're proteins. Because if you're going to attach RNA polymerases to the DNA, well, think how many different places that is in your DNA. So you need lots of different transcription factors to do it. So some of them are activators and are involved in starting transcription. Some of them are repressors and are involved in stopping transcription. So I remember this PowerPoint. This is world-class animation at its best. Are you ready? Let's watch the transcription factor in action. Whoa. Firstly, this bit of DNA here, this is a eukaryotic cell. That is not an operon. But even in a eukaryotic cell, we have these promoter regions. Now, these promoter regions will be right next to the gene that you're trying to transcribe. Actually, that's not always true. What I probably should say is the promoter region is actually right next to the gene you're trying to transcribe in location, not necessarily right next to it in the DNA sequence. You have to remember, your DNA doesn't just sit like a flat line in your nucleus, does it? It's twisted, it curls, it even maybe is wrapped around a histone protein. So what I'm saying is, yes, it might be physically right next to it in terms of base sequences, but it may be that its DNA is actually curved in on itself, and the promoter is the other side of the DNA, but the way that the DNA curves actually makes the promoter physically right next to the gene. So from a locational point of view, they are very close. They're not necessarily close along the DNA sequence. So transcription factor comes in. This one is binding to the promoter, and it's attached RNA polymerase to the promoter, and my gene is transcribed. That, ladies and gents, was an activator, wasn't it? It started transcription. Well done, promoter. Look, there's messenger RNA. Oh, I didn't have the budget to let messenger RNA leave the nucleus. Sorry, guys. I enjoy the half-hearted attempt to get the nuclear envelope, by the way. I enjoy that. So, if that's how we switch it on, what about switching off? So when a gene is not to be expressed, when it's to be switched off, the site on the transcription factor that binds to the DNA gets blocked by an inhibitor molecule. So now, if we look at this transcription factor with the inhibitor, oh no, look, it just bounces off. Oh, it's now a repressor because it's stopping transcription. Now, this inhibitor doesn't necessarily have to bind transcription factor. It could bind to the promoter. Either way, if you're stopping transcription, it's acting as a repressor. And this little yellow bit is just clarifying what I said earlier. Many genes have promoter regions some fair distance away, but because of how your DNA can bend, spatially, the promoter region will still be right next to the gene. So some nice examples of transcription factors. Probably the most famous ones are involved in regulating the cell cycle. So G1, SG2, and the M phase. The ones you might hear about are tumor suppressor genes and proto-oncogenes. These are genes helping regulate cell division via transcription factor, and it is those genes which get mutated. For example, proto-oncogenes can be mutated into oncogenes, and that can lead to uncontrolled cell division or cancer. Apologies for the sad reminder. But yeah, this is just one of those things that these transcription factors are heavily involved in. So these tumor suppressor genes, by the way, they literally are coding for transcription factors, and transcription factors are involved in regulating. Okay. Look, another example. Now this is not spec specific. This is just a cool example of a steroid hormone doing it. So estrogen physically stimulates a transcriptional factor. So because estrogens are steroid hormones, lipid soluble, it diffuses straight across the membrane by simple diffusion. And it's a complementary shape to the shape of the receptor molecule. So when it binds the transcription factor, it releases the inhibitor, which then allows the transcription factor to bind to the promoter and transcribe the DNA. How's that for cell signaling? How efficient is that hormone? So again, without estrogen, we've got this normal situation of the transcription factor having an inhibitor. So the gene, I'm going to presume this is a gene to be involved in steroid action, so some sort of building, whatever that might be. Maybe it's a gene involved in respiration to increase respiration rates. Don't know. But when estrogen is present, diffuses into the cell, binds to the transcription factor, changes its shape, so it kicks out the inhibitor. Boom. There we go. So that's eukaryotes and the idea of transcription factors binding to promoter regions and either switching on or switching off that gene. So next thing I'd like you to do goes back to the lac operon. So this is what you did last lesson. I am well aware that the lac operon there's a lot of new stuff and it needs time to play. So what I'm asking you to do, and I've already put it on Teams for you. So this is on the 13B channel of our Teams. Please click that link and it will take you, I wonder if it lets me go in. It'll take you to a little Neopod lesson where I've put 3 tasks, so things to do on Neopod. And I've made sure they're tasks that get marked. Optional nickname. The goat. Like biology goat. Actually, no, that's Mrs. Hall, isn't it? So the first one is can you draw the lac operon without looking at your books, your notes? You're going to have 2 minutes to do it, and I've given you a bit of help with actually what sort of genes to do. This is where I wonder if my pen actually works. Hello. Anyway, submit. And the next one is a matchy match. Match the lac operon component to its function. So you just sort of click one. Oh, what could that be? What's the structural gene? Maybe it's coding for oh, damn it, I'm wrong. And the final one is a big fat fill in the blanks. All the blanks are here at the bottom, and you sort of just drag them up to the top. Oh man, I got 0 out of 17. Never mind. Anyway, so have a play with that. Hopefully that should help you, remind you about the lac operon and feel a bit more confident about it. And when you're ready, let's do this next bit. So, what about when you've already made the messenger RNA? How can we alter the messenger RNA to affect gene expression? So that sentence there is literally the core of this. Post-transcriptional level regulation is where we actually edit the primary or the pre-messenger RNA and the removal of introns to produce mature messenger RNA. So what this is saying is that when messenger RNA is first made, so I'm going to call it baby messenger RNA because it's just been made, just been born. Obviously that is not scientific. We've got the term primary, or we're all going to see it as pre-mRNA. Baby mRNA is not going to work. Not gonna work. Not going to work. This pre-messenger RNA isn't actually going to code for the right protein yet. So we need to turn it into mature messenger RNA, which is then going to be able to go to the ribosome to be translated. So, ooh. And then we've got this word, a removal of an what the monkey is an intron. So let's look. Look at you got definition straight away through an intron. Oh, nice. An intron, a non-coding region of the DNA. So it's a sequence of nucleotides within a gene that doesn't remain in the messenger RNA transcribed from that gene. So in other words, it's part of the gene, but it's not actually coding for amino acids in the protein. It's a non-coding region. So it's something that needs to be removed if you are going to translate the correct protein. So what I'd like to do, just pause with me for a second as you do this little task. You can see right there is another definition we need. It's called an exon. If an intron is a non-coding region of DNA, a sequence of nucleotides within a gene that does not remain in the messenger RNA transcribed from that gene, what do you reckon an exon could be? Just pause me. And I presume you're back because you've done that. An exon is the opposite. An exon is a coding region of the DNA. A sequence of nucleotides within a gene that does remain in the messenger RNA transcribed from that gene. So exons will go to the ribosome and get translated into proteins. Introns, non-coding parts within the gene, will not. The way this works is the idea of splicing. And the enzymes that are involved in this editing, in this splicing, are called endonucleases. You may have heard of them before. Endonucleases, physically chopping up parts of the DNA. If you've not heard them before, you'll probably hear them again when we do genetic engineering. So the introns are the green ones, the exons are the orange ones. So if that's the DNA, we make our single-stranded pre-mRNA. Notice it's exactly the same, but it's a bit thinner because obviously it's now single-stranded. And yet the only parts that go to the ribosome as the final mature mRNA are the exons. The green introns get spliced out. Think a big pair of scissors that these endonucleases are doing as enzymes. They're like a big pair of scissors chopping up all the introns and then splicing together the exons. So obviously there are other enzymes involved in like joining them back up again. Isn't that fun? And it does actually say down here that we still actually have no clear understanding of why we have introns in our genes. Clearly there must be some evolutionary advantage, otherwise why are they there? And often they are multiple repeats. So just like the same thing repeated again and again and again and again and again. I know we're not there yet, and I know your focus probably at the minute is, oh my daisy, I've got an exam on module 5 and 3 and things like that. But this idea that there's multiple repeats will come up again when we look at DNA sequencing and sort of the forensics side of it. Because we have multiple repeats in our introns, I can work out who you are. I can compare you to your mummy and daddy. I can play a Jerry Mikhail game of who's the daddy. Anyway, let's play with this. So we have a template strand of DNA. Can you write down the pre-mRNA that would be coded for by that template? So right, pause me and then start me again when you've done. Okay, let's see if you got it right. Use a green pen. Did you all? Just complementary base pair it. That's a template strand, so the pre-mRNA is just obviously just the opposite. So it's called pre-mRNA because it still contains the non-coding regions of DNA. It still contains all the introns. It still contains the introns. So if we now say that this is the actual gene, and we know that this is the exons, these are the exons, so just it's probably not, it's a bit better if I do it like that. Exons are there, introns are there and there. The actual base sequence will be exactly the same, but it won't have any of the introns, will it? It'll just be the 3 exons joined together. Nice little splicing animation. Let's see if it works. I think it still does. I'm getting to that stage in my career now where all these animations I find, because you have to bear in mind, like proper lesson planning and like when I used to stay up till midnight getting every lesson because I've never taught it before. Those days are nearly long gone. So where is it? Oh. Messenger RNA splicing. That'll probably do. Don't remember it looking like this, but here we go.
Once a gene has been located and transcribed into mRNA, it must first be edited before it can be translated into a protein. This editing process is called splicing. It involves removing non-coding regions called introns, leaving only the protein-coding exons. In the cell, the introns are removed by special enzymes which recognize specific sequences. These enzymes cut and rejoin the coding regions for translation into protein.
So I don't know, I generally do not know if you heard that, if the sound was on for you, but that was it. That was it sort of showing you just the idea of these endonucleases sort of just stopping at an intron and then letting the intron wind and then splicing at the end of that intron with the enzyme and then reforming. So you're just left with the exons. Okay, final part of this super hard spec point. We've covered transcriptional level at both eukaryotes and prokaryotes. Prokaryotes was the one with the sequence, the lac operon, the step-by-step. We've looked at post-transcriptional level. Post-transcriptional level is the removal of introns, so pre-mRNA becomes mature mRNA. The final thing to look at is post-translational. Do you know what the great thing is about this? You've already done it. Those who were paying attention during Module 5 when we looked at different hormones like adrenaline and how the idea of a second messenger works and what cyclic AMP does, then you already know what happens here. You already know. So some of the proteins you make by translation are not functional straight away. Some of them are in a... like my brain says instantly said inhibited state, but that's not actually what we call them, is it? They're in a dormant state. There is a word, and it's going to come to me. For now, I'll just say dormant. They're not functional straight away. They have to be activated by something in order to function in metabolism. So the cell basically needs to be told, and if the cell needs to be told to switch on your protein, then that's going to be cell signaling. And it's very likely going to be a hormone. So a lovely example of cell signaling, as we know, is cyclic AMP, where the hormone binds to the receptor on the cell surface membrane. You might then get, again, there are different ways to get cyclic AMP, but it might be this one. It might be the activation of a G protein, which then activates the enzyme adenyl cyclase, which then turns ATP into cyclic AMP, and then cyclic AMP goes around phosphorylating things within the cell to activate them. A nice example is protein kinase A. So protein kinase A is an enzyme made of 4 subunits. When cyclic AMP is not bound, they are together and inactive. When cyclic AMP does bind, it changes the enzyme's tertiary structures and actually releases 2 of the subunits. These 2 little subunits are now actually active to go around the cell and catalyze reactions. That is translational level gene control. Because your actions, in this case it's a cyclic AMP from that hormone that bound, or it might not be a hormone, it could be like another type of molecule. I don't know, even could be like a neurotransmitter, could be like those molecules involved in, like interleukins and cytokinins involved in infectious disease. I mean, they're not hormones, but they function the same. Well, function the same when they get to the cell, if that makes sense. Switching on, switching off things. So that's just a really nice example of what it could be. That is translational control. And if you do want to look at this in all the depth, if we really want to look at it, and then there is that. It does. I think this is from the textbook. But all this first bit, that's we know all this step 1 to 5. That's boom. That's hormones. That's what adrenaline does. It's no different there. So the signaling molecule, such as in this case is glucagon, binds to a receptor of the plasma membrane of the target cell, activating the G protein. G protein activates the adenyl cyclase enzyme, and the adenyl cyclase enzyme turns ATP into cyclic AMP. And cyclic AMP activates enzymes, proteins within the cell. This example was protein kinase A. When protein kinase A is activated, it phosphorylates proteins, hydrolyzing ATP in the process, including another protein called CREB. And CREB is a transcription factor which enters the nucleus, binds to the promoter region, and switches on DNA to translate a brand new protein. So we've got this lovely link of post-translational control leading to transcriptional level control of your genes, which I love as an example. So anyway, it may be you want to just repeat that bit. I mean, just play me again. Like, the more it goes into your brain, the longer it should stay there, shouldn't it? So thank you very much for your time. Hope that went okay. Hopefully see you all next week. Bye bye. Oh wait, there is a place. I don't think there's any like specific questions in the in your booklet, but there is a space in the booklet to get notes and things down should you wish to do that. I'm not going to force you to because it is personal preference how you do your notes, but there is a space in the booklet. Anyway, bye.